Hello everyone, I'm Masaf Nabashi from Sony CSL, and today I present our CSDC laboratory Open Systems Exploration for Ecosystems Leveraging. One of the most important global agenda the world is facing today is the sustainability of social and ecological systems. A pioneering work in systems dynamics modeling in 1960s, the Club of Rome report already raised high alert. A global collapse of our heavy industry civilization is anticipated to occur by the global increase of population and depletion of natural resources. Half a century has passed, and recent study confirmed the plausibility of this simulation. It even showed that decades of effort towards the conversion of technology and society to a sustainable path was not producing significant effect to escape from the disastrous trajectory. An independent review from ecologists also warned that there will be a global collapse of biodiversity and its associated ecosystems functions that would threaten human well-being. The study is clear to determine the cause as the increasing pollution from human activity and most globally environmental load arising from agriculture such as soil erosion, onslaught of fertilizer spill in water systems, and chemical pollution. When considering agriculture, the global bearer of the world food production is not a large-scale agribusiness industry. Actually, 70% of the planet's food is produced by smallholders, family-owned small-scale farmers who cultivated 2 hectares or less. They work on 80% of the world's arable land, and with 500 million family farms, make up 1/3 of the world population, which is the most widespread form of farming. The economical incentives in developed countries, however, tend to converge large-scale agribusiness into environment-destructive monoculture, which inherently produce pollution and overconsumption of fossil fuels. Health problems such as obesity is also associated with the concentrated production and distribution of modern agriculture. Sustainability initiatives from third parties such as United Nations reports emphasize that we should support smallholders, small-scale, locally distributed, and diverse form of agriculture in a more ecologically sustainable way. Promotion of biodiversity and integration of widely untapped edible plant resources into local production systems is considered as a crucial premise for sustainable agriculture. The enhancement of smallholders' practice, promotion of biodiversity, and vitalization of sane local economy is the matter of total optimization of social ecological complex systems. This objective clearly calls for a bottom-up citizen movement, gathering multiple stakeholders, as well as the design of self-organization and multi-scale optimization with strong support of complex system science and affordable technologies. Our e-laboratory aims to provide a prototype of boundary institution acting to resolve these problems, which basically create and provide information related to sustainable primary industry and function as a hub to connect citizen and scientific communities towards the realization of open source distributed food production systems and the augmentation of other ecological capital. How could it be achieved? A transversal strategy linking the projects in our e-laboratory is to consider important factors of biodiversity as an interface between human and nature. By combining actual field observation and ecosystems agents such as plant and animals with computational databases and models, we aim to augment humans' ecosystem function and services. If the increase of human population is inevitable, we need to realize the ecosystem where the population growth entails constructive meaning in ecosystem functions. Improving our function within the ecosystem, increasing the benefits we provide, must form the foundation of any viable kind of sustainability. This approach is based on the open system science in contrast to conventional science of closed systems. Traditional science put importance on the reproducibility and predictability in a confined and simplified model such as laboratory experiment, which we call closed system. Open system science tries to tackle the non-reproducible real-world problem with its whole complexity. It forces us to act as an internal observer, struggling to manage the system without halting the operation and without isolating the subsystems. For more details of open system science, please refer to the invited talk of Professor Mario Tokoro in the E-session Open Systems Exploration. The initial member institute of the e-laboratory consists of 3 research institutions from 3 countries, in which participants pursue their own project ranging over agriculture, ecology, food science, and related theoretical aspects of complex system science. In 2014, 6 initial projects have started in open collaboration inside and outside of the e-laboratory platform. Each project will provide databases, tools, and tutorials as a part of the research and education program of CSDC. Let's briefly introduce each of the 6 field laboratories. Culture Project by Masafu Nabashi. Culture Project aims to construct a novel system of small-scale biodiversity-based agriculture with open complex systems perspective. We aim to optimize the culture in the level of synecology, or the ecology of community, where various plants and animal species interact under competitive and symbiotic relations. We practice in several experimental farms and family gardens in Japan, high density of mixed culture that generally reach over 200 species in 1,000 square meters. With no input of tillage, fertilizer, no chemicals, the vegetation follows complex self-organization process of natural niche, usually referred to as ecological optimum. As a summary of our activity until 2014, we evaluated the biodiversity of introduced species in the monitored culture fields. In a surface of only 0.3 hectare, our 250 edible species grew and produced a wide variety of yields. This species diversity is greater than traditional sustainable agriculture, renowned for the diversity of crops and high biodiversity conservation value. This means culture can realize a regional scale edible species diversity even at a very small scale. In order to manage the extremely high diversity and complex behavior of natural niche formation, we construct field databases with biodiversity and related information. We develop complex network interfaces for the reconstruction of ecological network in the field. The management model also requires the web-based network interface, which is important for the sharing, evaluation, and discovery of farming options. These databases were used to refine the farming option, including a collaboration with Open Systems Data Analytics. Open Systems Data Analytics by Sasaki is the next project. The data related to ecology and agriculture are exponentially increasing, such as biodiversity database, monitoring data of agriculture fields, buying history of products, nutrition profile, geo-environmental assessments, and so on. Such big data is gaining greater importance on the future of human society. In many cases, they are open systems, and we cannot rigorously determine causal relationship and obtain full control of it. Still, in such open and limited situation, inference of causal structure is useful and needed for the management from inside of the system. In Open Systems Data Analytics, we're trying to develop novel analytical tools of massive data that have many variables dependent on each other using probabilistic graphical models and methodologies of physics. We developed a protocol of open systems data analytics based on the Tokoro's procedure of open systems science. As a concrete methodology of causal inference, we adopt Reichenbach's principle of causality and use the combining stage algorithm to infer the causal relationship. The first step is to define the problem and its initial domain to start the exploration. Then we set initial variables that are supposed to play an important role and start to get their data, even partial and incomplete data, as far as possible. Here we've made an expansion of the initial model with human exploration. We should repeat this procedure until we can achieve sufficient level of understanding of the system. And by keeping up with the renewal of the model, we can proceed to predictions, interventions, and construct theories if temporary consistent structure exists. This forms a standard protocol of open systems data analytics. We applied this protocol to infer causal relationship in the biodiversity database of Sineco project. This database consists of biodiversity and farming knowledge records observed by citizens in several Sineco fields and surrounding environment in Japan from 2011. It contains more than 20,000 pictures and associated taxonomical information of more than 1,000 plant animal species. We study the causal relationship of co-occurrence of species, date, and place information in the database. As an initial step in operation, we apply the protocol 1 to 4 of open systems data analytics. This is a visualization of the results. It describes the causal structure inferred between the nodes that correspond to species, their observation date, and place. The red arrows are the causality suggestions unknown to the experience or other literatures. Therefore, red arrows are candidates for further observation. The blue arrows are the causality suggestions that are already known to the experience or other literatures. Therefore, blue arrows are validation instances of the model. By comparing the number between red unknown suggestions and blue known suggestions, we can calculate the information of known suggestions for further exploration. In this figure, the orange circles depict the amount of information that unknown suggestion could bring as a reward of prediction if the suggested causality was true. Therefore, the high information domain in the red circle is a preferential candidate that merits to be explored in the next step. We expect to repeat and integrate this analysis year by year to investigate the degree of achievement in understanding ecosystem dynamics of Sineco field. The next project is Peer-to-Peer Food Lab, P2P Food Lab, by Peter Hanap. The goal of the P2P Food Lab project is to develop innovative and sustainable solutions for the growing, harvesting, distribution, and disposal of food. The first phase of the P2P Food Lab project is to design an online-offline platform that groups food growers, technologists, scientists, and citizens that can be used to study alternative solutions for food production using a bottom-up approach. In our first experiment during summer 2014, we developed a small internet-connected greenhouse. A sensor box was placed inside the greenhouse that took daily images of the crops and measures the air temperature, air humidity, and sunlight. The sensor box was built using standard off-the-shelf components such as Arduino, Raspberry Pi, and standard webcams. The goal of the connected greenhouse was to create an online social network of participants and gardeners and evaluate the use of sensors for the study of crop growing and support new modes of crop production, in particular small scale of microagriculture. This summer, we continued this work in a new experiment called Citizen Seeds. In this experiment, we gave participants a collection of seed of 18 varieties of vegetables that they had to plant on a square meter plot. We also gave them planting schedule that indicated when to plant the seed. The shared seed collection and calendar help in aligning the participants, in comparing the data, and in stimulating social interaction. Participants are asked to upload photos of their plot and the vegetables once a week. To measure the environmental data, including sunlight, air temperature, and soil humidity, the participants had the option to use an electronic sensor device. On the website, the participants can compare the evolution of the plot and the climate data. This experiment was the first of a series that aims to involve many amateur gardeners in scientific experiments related to food growing. Topics of interest to be studied include analyzing the effects of crop intermixing, measuring the evolution of soil biology, tracing the phenotypical adaptation of varieties to local conditions. In the long run, we want to use the platform to involve communities in the development of new technological tools and sensors to monitor and optimize alternative sustainable cropping techniques. The next project is the Bee Laboratory by Anne-Marie Mas. The Bee Laboratory. Honeybees are bioindicators. They provide a constant stream of information on the environment in which they forage via their daily activity and via the pollen and nectar they harvest. Environmental problems such as the use of pesticides can be detected by monitoring the colonies with audio and video tools and by scanning their daily activity over several years. To study the bees in their natural environment, following the footsteps of Von Frisch and other ecologists, we have built a customized sound device. The Sound Beehive experiment monitors the development of a bee colony on the basis of the sounds it generates and creates artistic expression mediated by artificial intelligence. The Sound Beehive is installed in the Urban Bee Laboratory on a rooftop in the Brussels city center. We equipped an existing Ware beehive with sensors, microphones, and cameras. Data is streamed to central repositories and analyzed using statistical techniques and graphic visualizations. Art exhibitions are set up for public awareness raising. The art installation at IGCAI 2015, held in Buenos Aires, brings a human-sized version of the Sound Beehive. The video shows an artistic rendering of AI analysis of colony behavior, combining real audio data with measurements of the microclimate inside the hive, temperature, CO2, and humidity. A pollen database is created to monitor the foraging areas. By studying the pollen in a sample of honey, it is possible to collect evidence of the geographical location and genus of the plants that the honeybees visited. As such, we start to trace green corridors through the city. The next project is One Health Food Lab by Kaoru Yoshida. Under the slogan of One World, One Health, our One Health Food Lab scientifically examines foods and medicinal herbs which were produced in different growth conditions and placed in the market to evaluate their quality and safety to the health of human and other creatures here in the earth. In 2014, we studied functions, compositions, and tastes of common vegetables in relation with their farming methods. Through the studies, physiological differences of plants related with their growth environment and their effects on human health as foods and drugs have been partly elucidated. The next project is Open System Simulation. by Takahiro Sasaki. In this project, we pursue a new form of simulation required for the management of open systems. As a specific instance of open system simulation, epidemics is an important factor of social ecological systems that threatens productivity and sustainability. We have been developing an integrated simulation of infectious disease, especially targeted for influenza. Influenza is a typical example of open systems where various aspects are all interrelated in a complex manner. Infection process, social system, geography, ecosystems, and so on. Therefore, you cannot understand the essential phenomena by just looking at each component separately. In conventional science that focuses precisely on a closed domain, we can investigate individual facts such as how the pathogen is structured, what is the micromechanism of infection, which genes are related to which processes, and so on. By limiting our focus on closed domains, we can accumulate this kind of elemental knowledge rather easily, which is however reproducible only in laboratory's environment. On the other hand, if you want to understand what happens in the real world and to obtain essential understanding of the phenomena, we cannot limit our focus just only on pathogenic mechanism. But we have to consider various factors such as interaction between pathogen and host, social structure or dynamics, natural systems including ecosystems and other hosts, geological constraints, and so on. One of our findings obtained through our research is on the phenomenon called limited diversity. This is an interesting evolutionary behavior observed in the real influenza viruses. While continual changes accumulate in the viral genome as time passes, there is always a chance for a new genetic lineage to diverge, which could theoretically result in ever-increasing explosive diversity. However, in reality, the diversity of the existing viruses is relatively limited to a certain extent at the population level and at any particular point in time. To provide a possible explanation for this limited diversity, we have built a multi-layered model which interconnects macro-simulation of epidemic circulation among hosts and micro-simulation of viral evolution driven by immunological interaction among hosts and viruses. As a result of interaction among immunological factor and microstructure of viral genomes, an advantageous region for viral survivability, what we call as window of evolution, appears in fitness landscape, which restricts viruses from evolving explosive diversity. This insight could not have been obtained without integrating multiple factors and taking them into consideration at the same time with the platform of open simulation. This is the conclusion. With 6 initial projects, our e-laboratory gathered scientists, engineers, and artists to tackle the sustainability issues in food production and ecosystems management, ranging from urban gardening to agricultural production, from on-the-fly exploration to integrative simulation, from human to environmental health, from data analytics to artistic expression. Sensing technologies, databases, analytics, and citizen science will be combined in order to realize the leveraging of ecosystem services via human activity. By exploring ecosystem agents as a symbiotic interface between human and nature, this challenge would consequently be led to develop diverse, parallelly distributed practices of sustainable food production and related scientific domains. Our challenge aims to yield the potentials of self-organizing nature of ecosystems with a bottom-up organization of each project. Our research and education program consists of an integrated body of databases, tools, and tutorials which together elaborate research projects and citizen participation. Each project will share the materials with emergent collaborative process with others. And we hope this e-laboratory would serve as an open and pioneering platform that augurs the real citizen science addressing crucial sustainability issues. For more information, please visit our e-laboratory website. Thank you for listening.